Okay, hey guys, my name is Ben Siranosian, and I'm currently a PhD student at Stanford, but I'm presenting some work that I did last year when I was working at the Broad Institute. The title of this talk is Genetic and Transcriptional Instability Alters Cancer Cell Line Drug Response. And so I just want to start with the acknowledgments for this because this was a huge group effort across the Broad and really beyond to other places in Boston as well. And Uri Ben David, who's up here, really led this effort and kind of brought together a huge group of talented people on both the wet lab and computational side. You can see all their names here. And our two advisors were Ramin Baruchim and Todd Golub. And this work was funded by the Broad Institute. I received a travel fellowship from the ISCB and also from Stanford to come here and present. So back to the science. Today we're going to be talking about cancer cell lines, right? So immortalized cell lines that can proliferate, perhaps indefinitely, in a dish after being removed from a human patient. And cancer cell lines have been used for biomedical research for years now, often for preclinical drug screens and trying to identify new chemotherapeutic compounds. However, there's been a kind of crisis of reproducibility in these drug screen results, where a recent study found that over 50% of preclinical results based on cancer cell lines could not be reproduced, some of which due to the actual cell lines themselves. And this is a huge, both money, time, effort, et cetera, problem within the biomedical research community. So my work is trying to address what the actual source of this failure to reproduce is, and we're going to kind of break down the talk into three sections today. So first, how variable are isolates of the same cancer cell line from different sources, different labs, different time points, different groups? Is this just neutral genetic variation, or does it actually matter for something like a genetic drug screen? And is it all bad news? What can we do about it? And what do we learn about cancer evolution more generally by looking at this data? So just to give you the results, first off, isolates of the same cancer cell line are very variable. This is perhaps unsurprising because they're cancer. They're always evolving. They're always changing. And we're not the first people to publish on this or to kind of find this out. Does this variation matter? Yes, it does. We show a really kind of striking difference in response to chemotherapeutic drugs based on the genetics of the strain isolate itself. And it's not all bad news. We can actually do something really cool with these isogenic-like cell lines. And we kind of provide some ways that you as a researcher can best deal with the problem that this presents. So first, we're going to look at the genetic and transcriptional variation of multiple isolates of the cell line MCF7. And MCF7 is a estrogen receptor positive breast cancer cell line. It's widely used throughout biomedical research. And we obtained 27 different strains, supposedly all normal MCF7. 19 of these strains were wild type. 7 had neutral genetic manipulations, such as introduction of GFP or Cas9 or something. But you don't expect that to really change the underlying landscape of the cell line itself. And as a outgroup or kind of control for what we expect to be very different, we have one MCF7 that was placed in mouse. It was subject to anti-estrogen therapy and then removed back and repropagated as a cell line. So we expect that to be very different, but still kind of similar to MCF7. So these 27 lines were obtained. You can see on the right this kind of chart where the lines came from. They came from over 11 years from 7 different sources. These are both commercial sources, neighboring labs, freezers that we just kind of begged people to let us take their MCF7 from. And we had this really great collection of actually 11 strains maintained by the same lab group over 10 years. And they were subject to many freeze-thaw cycles, but they weren't proliferated or passaged too often, I think. So first off, we're going to look at the genetic variation. We did low-pass whole genome sequencing on these 27 strains of MCF7. And on the right is a heat map where you see losses of the genome indicated in blue and gains of the genome indicated in red. And we've just kind of done a hierarchical clustering of these 27 different strains. And as you expect, right, this is a cancer cell line. We see about a quarter of the genome was either lost or gained on average. And strain M here is our outgroup. You can see it's way on the left. It's definitely an outgroup in the clustering, and that wasn't included in any of the statistics I'm talking about, just so you know. 20% of the copy number variation was common to all strains. 10% was unique to an individual strain. But most was really variable across the entire population. Some gained, some lost, some were neutral. If we look at the clustering, one interesting thing that we saw was the 11 strains from the same lab. They're all colored in blue up there on the right of the clustering, and they're all very similar to each other. They were obtained 10 years different between the first and the last, but they weren't passaged, so that'll come back as being very important. Strains D and E are very close together genetically. They're actually maybe 1 or 2 passages apart, so that's what we'd expect. And as I said earlier, M is an outlier because of that in vivo expansion that they did in mice. We then did some targeted deep sequencing of cancer-related genes, around 400 or so. And here in this heat map, once again, I'm showing only genes that were differentially mutated across 1 or more of the 27 strains. So nothing is going to be constant. That's shown in this heat map. And yellow indicates that there's a mutation in a particular gene. You don't really need to know what the genes are, but just kind of showing that, once again, we see correlated changes in the mutational landscape of these 27 different supposedly identical MCF7 clones. To look at the gene expression changes, we used what's called the L1000 assay. It's basically a cheap, efficient way to profile the levels of 1,000 gene transcripts. And from that, you use a big linear model to infer the rest of the transcriptome. It's like RNA-seq on the cheap. It's really well correlated with RNA-seq. We found that the expression profiles from these 27 MCF7 strains were best correlated with MCF7 and other breast cancer cell lines that we had profiled previously. So they're still very close to what you'd expect. But if we look at pairwise comparisons between one strain and another, there was a median of over 600 genes differentially expressed at a certain log-fold change of 1, between pairs of the same strain. So that's 650 genes differentially expressed between identical MCF7 strains. So by now, I hope I've convinced you that there is a huge amount of variation in these 27 different strains. They're supposedly the same thing, but is that just a curiosity, or is it actually something that's going to impact your research, either if you're doing a drug screen or you're looking for new candidates for something? And the answer, we believe, is yes. I'm going to show a few vignettes of kind of the interesting things that we found. So first off, MCF7 is used as a model of estrogen receptor positive breast cancer. So you expect any MCF7 clone you get to both have the estrogen receptor gene and to actually express it, because that's the model that you're trying to use. And here on the top figure, we're looking at the copy number of the actual estrogen receptor gene. And you can see that it's very variable across the 27 different strains that we obtained. Some gained, some lost, some are neutral. And we actually show that the level of transcript down at the bottom is well correlated with the copy number level of the estrogen receptor gene. So this would be a big problem if you're screening a compound, for example, that is active against the estrogen receptor, and your strain doesn't express that gene at all. That would be kind of a false result. Next, we looked at a panel of over 300 drugs that are potentially active against cancer cell lines, and we tested them at a single concentration across our 27 lines. This includes known chemotherapeutics, inhibitors of certain pathways relevant in cancers, a couple antibiotics, some random stuff. Not necessarily important, but we saw 55 compounds that were active in at least one strain. That's defined as growth inhibition greater than 50% in at least one strain. And on the right here is the heat map, where a darker blue color indicates a much more sensitive interaction between the strain and the particular drug. However, among these 55 compounds, 48 had at least one strain that was entirely resistant. So you'd be treating, 50% of the lines would be sensitive, 50% would be resistant, and there's something clearly going on with these drugs. To kind of follow up on that, we took a few drugs and did a much more in-depth 8-point dose response testing, where you profile the viability as you increase the dose of a certain drug. And here on the right, I'm showing the 2 most sensitive and the 2 most resistant clones to a drug called bortezomib, which is a proteasome inhibitor. And once again, as you can see, we have vastly different drug response curves for these different MCF7 clones. We were able to show that actually the drug response itself is well correlated with the expression of certain genes in a pathway relevant to the mechanism of action of that drug. And I've shown that for just a couple examples here. Strains that are sensitive to cell cycle inhibitors are upregulating the cell cycle signature, as shown by GSEA, gene set enrichment analysis. And that shows cells that have kind of become addicted to a certain pathway through their expression are more sensitive to inhibition of that pathway. And you can kind of show the same thing for certain classes of mutations where having a mutation in that gene makes you more sensitive to a drug inhibiting that pathway. Shown here for PTEN and AKT, both important members of a growth and development pathway for cancer. So finally, I just want to kind of tie things up and show you a few recommendations that we give for any scientists that are working with cell lines. First off, is this a fluke in MCF7, right? We have a single cell line. Was it just a weird cell line or something like that? We did the same experiment again in A549, which is a commonly used lung cancer cell line. And we found 23 strands this time, both wild type and genetically manipulated. And we saw similar levels of molecular and transcriptional variation across this panel also. So it's not just not just isolated to MCF7 or breast cancer cell lines. And then thank you to a reviewer, we did deep targeted sequencing of 11 different cell lines, which took a long time, but we did it. And we showed that MCF7 and A549 are kind of just in the middle of the range of genetic variation for this increased panel. So really, this is not confined to a particular cell line. This is a problem more generally. And what drives this genetic variation? Because we had a time course and samples from different labs, we were able to show that freeze-thaw cycles, so taking your cell line out of the freezer and maybe drawing aliquots from it, samples from it, is not really that important for changing the genotype. However, passaging your cell line many times and supposed neutral genetic mutations are going to have a much larger consequence. Finally, is it all bad news? I like to say no, because now that we're more aware of this sort of neutral genetic variation that's happening, researchers can profile their cell lines ahead of time and actually make sure that what they receive from a vendor or from a neighboring lab is actually the strain that they expect. And it's actually kind of useful because strain panels like this are basically isogenic cell lines that are modified in only a few key genes. And you could use something like that to identify unknown mechanisms of new drugs. And through a kind of test that we did, we were able to actually show that a strain panel like this can identify the mechanism of action of new drugs better than a cell line panel, where you're much more variable. And with strain panels like this, you can start to do some more research into cancer evolution more generally, such as are there competitive interactions within a population that favor certain mutations. But we haven't done any of this. Finally, we kind of have three main conclusions for researchers working with cell lines themselves. We've shown that inconsistencies in drug response or preclinical testing may be due to the underlying genetics and transcriptional variation within these cell lines. And as a researcher, you really need to confirm the identity of any cell line that you receive. Thankfully, any sort of low-pass whole genome sequencing is a cost-effective and easy way to do this, because we've shown that genetic distance in terms of whole genome sequencing can really well correlate with transcriptional and drug response also. And you kind of need to go back and keep doing this after you transform your cell line or you've put it through a few passages, because we know that these things are going to change. To help researchers do this, actually, we created a tool called Cell Strainer, which is available online. It's currently in beta, but it can take low-pass whole genome sequencing and compare the genetic distance to a bunch of references and say, how close are you to all these other cell lines? And be on the lookout for this when it comes out in publication. That's all I got. Thank you guys for listening. And once again, acknowledgements, because these people are great. Yes?
So do you have a way to determine how much of this phenomenon is because of mutation versus how much is because different subpopulations, original cell line gets amplified, some subpopulation gets eliminated during the passaging?
Yes, yeah. So we did single cell clones also to kind of try to get at that, and we took single cells. Oh, repeat the question. Sorry. How much of the variation is due to pre-existing populations, I think is your question. And to answer that, we did single cell clonal expansion, where we took a single cell from this strain and expanded it, and we showed that the majority of it is kind of just naturally occurring. It occurs as the cell grows and divides. So we believe that there's some evolution or some pre-existing variation, but a lot of it's just kind of occurring. And it's not really correlated, like what happens, as you expand 10 different clones. They don't really go in the same direction.
I have a question.
Yes.
So I think it was really cool. It's a really cool study. One comment is because I come from a clinical background, so it's really important to use controls, and MCF7 is used as a control cell line in repeated clinical testing runs for patients.
Mm-hmm.
And so making clinicians aware of this, that there could be variability, and using something that has extensive variability as positive control in clinical testing. I think it's really important to be aware of it, because most of these people are pathologists who are doing all of these clinical testing, and they're not aware of this kind of variation that we see in the background. My question is, have you looked into if we could control this variation in any which way? So are there certain features which are always variable, and can we take them out when we are analyzing the results from these cell lines? And also, is there a way, did you do any experiments to see if all of those three factors that you showed, if we did it to any of those, like if we could control for them, do we still see that much of variability?
Okay. Comment about the fact that as clinicians, you also need to be aware of this because they use MCF7 as a positive control, I believe, in a lot of studies. That is definitely true. I think the good thing is that if you are consistent and you haven't manipulated the cell line too much, at least within a lab, your results will be consistent. Kind of what we showed for that panel from 10 years in the same lab. Can you do anything to remove this sort of variation or be aware of what's happening? I think the answer to that is sort of no, because the changes aren't always happening in the same genes, in the same copy number of locations, right? It's kind of a stochastic genetic variation. So so it's pretty random. I think the only thing that you can do is periodically check and periodically confirm what you have.